everyone. Welcome to the webinar we have with Affymetrix here. Today's topic is talk about how Nexus Copy Number analyze the data from the Affymetrix Axiom platform. So the format is we're going to have Mary Jo Galina. She's the marketing manager for Axiom product. Give an intro first, and then my name is Ziwei Che from BioDiscovery. I'm the director of application science. I'm going to follow up with the Nexus Copy Number part for data analysis. So during the presentation, if you have any questions, please use the GoToWebinar panel to type in your question. We are going to check the questions along the way. And also, for the at the end of the our webinar, we have a four simple poll questions to basically you guys can answer to give us some feedback. All right. So without further delay, I'm going to hand over to Mary Jo. Hi, Mary Jo.
Hi, Ziwei. Thank you. This is Mary Jo Galina, and I'm going to give you a brief introduction to the Axiom Genotyping platform before Ziwei shows you how the CNV capabilities work on the platform. So I hope everyone can see my slides. So the Axiom platform is an end-to-end genotyping solution. We can genotype arrays that are from our catalog, pre-designed arrays, or custom arrays that are built by the user with whatever content that they choose. The platform includes an Axiom reagent kit, a automated or manual protocol for target prep. The GeneTitan instrument is used to process the arrays and do all the imaging of the arrays. And then we have genotyping software, which actually makes automated genotyping calls for the SNP markers and indels. The platform comes in two different formats, the 96 format and the 384. The 96 has a higher number of markers on it because it has more real estate, and both of the formats use the same Axiom chemistry and the same GeneTitan instrument to do the plate processing. You can see that these arrays are like 96 arrays are processed in parallel and hybridized to separate samples on these different formats. So this can be done on our catalog arrays, or you can customize arrays to have specific content to your research interests. Of course, you can take one of our catalog arrays and modify it by replacing or removing, adding markers. And the manufacturing lead time is pretty short, 4 to 6 weeks. Our designs never expire, so you never have to worry about a bead pool expiring. You can always reorder the arrays anytime. It's sort of like printing books on a printing press. We can make your arrays for you at any time. The Axiom content is organized into modules, so it's nicely organized that makes it easy for you to choose what might be on a custom array. Or on the left here, all of these modules are actually part of the UK Biobank array. So this is just an example of how arrays are put together. We have, for example, GWAS panels for each of the 1,000 genomes populations. You can choose to put exome content with coding variants and functional variants or pharmacogenomic markers. We also have, now we have available copy number variants. And any or all of these categories and more can be put onto an array. It's all organized very nicely into modules. So here's an overview of how the CNV analysis works. The genotyping is done, as I said, on the GeneTitan, and genotyping files are generated by the genotyping console or Affymetrix Power Tools software. The genotyping data files are then input into a new software that we've created that's called the Axiom CNV summary tool software. And this is the piece of software that actually calculates B allele frequencies and log2 ratios for each marker for each sample. So this piece of software generates files that are compatible for upload into BioDiscovery's Nexus software, which Ziwei is going to show you in a moment. So I just want to show you how to get that software. You can download that from our website. So just go to the Affymetrix website and look for the Axiom CNV summary tool software, as well as a user manual that will tell you how to use it, and recommendations for settings in Nexus. Now, the brand new version of Nexus already has the settings as defaults, but should you be using an earlier version of Nexus, you may need to put in the recommendations that are in the document that's available on the download. So with that, I'm going to turn it back over to Ziwei, and he's going to show you some of the Axiom data in the Nexus software.
Thank you, Mary Jo. Let me get this to my side. I'm showing you. I have actually a PowerPoint presentation, and then followed by a live presentation of the software with the data Mary Jo sent over to me. So Nexus Copy Number software to analyze the Affymetrix Axiom array for copy number results. So to give you some background information about this software, Nexus Copy Number. Nexus Copy Number is our company's comprehensive and user-friendly copy number analysis software solution. It has been out since 2007. So right now, has been well accepted worldwide in over 400 research institutions and clinical labs. The typical applications using Nexus Copy Number range from research for constitutional sample or cancer samples, also people doing clinical routine cytogenetic lab work, doing case review, and then use Nexus Copy Number to identify any aberrations. It supports all Affymetrix arrays, including Axiom, Cytoscan, OncoScan, SNP60, 500K, or even 100K, all those old Affy arrays. So also supports NGS data or even other array platforms. The strength of the software, Nexus Copy Number, is that it can analyze thousands of samples in a single project. So ideal for Axiom plate data, as Mary Jo pointed out, the data you can have 96 well or even 384. So those data you get usually is a large batch of data. You want to do that kind of large data analysis. So Nexus was designed to handle so many samples in a single analysis project. It also has many advanced analysis functions after you get the copy number results. So the data has been loaded into Nexus Copy Number. So you get the results, but then a lot of people want to do research, and then based on the phenotype, the information or the clinical factors of the samples, you can do further data analysis based on the copy number results. It also supports any organism with the genome build. Here is just a partial list of organisms Nexus Copy Number can handle. So for example, human, mouse, rat, cow, cat, all those things include animals and plants and all different types of fish. I know a lot of people using Axiom to handle different species for the so-called custom arrays. Then Nexus can easily process them for copy number analysis. So here, just go over the workflow one more time. Mary-Jo already mentioned the workflow. Here, just go over this. The raw data from Affy scanner, you go through the Affy CNV summary tool, and from there you get the data imported into Nexus Copy Number. From Nexus Copy Number inside of the software, the first thing we do is to do the so-called data waviness correction. So a lot of time people found this data log ratio is wavy based on the GC content of that region. So the software can do the quick correction to straighten them up before you even go into a copy number analysis. So after that, the software has its own algorithm to call copy number segmentation from the log2 ratio and the biennial frequency information coming out from the summary tool. And once you get the segmentation done, you get a copy number results visualization. From there, you can actually visualize multiple samples or single samples, depending on what you want to do with your data. Besides the data, it also provides the QC score to say each sample, what the quality of the sample, how noisy the data is. You can go down to a single sample drill down to look at each sample individually, what kind of event that sample has. You can also do advanced analysis based on what you want to do with your data, depending on your research. Depending on your research, then we have so many different analysis modules you can use. The most common one people want to do research with their large data size, they want to locate the common aberration regions. So if you have, for example, similar samples, for example, tumor samples, you want to identify the common aberration regions, identify the copy number gain and loss regions. Maybe those are related to the oncogene region, and you want to easily identify from the large sample size. Also, based on the sample factors, you can do comparisons between different sample groups. You can also do clustering based on the copy number profile, how similar those samples are. You can cluster them into based on the similarities. You can also do enrichment analysis to find the functions for pathways, especially for research, depending on what kind of research you want to do. So those functions are all after you get the copy number results generated. The last one, at least here, is not a complete list, depending on really what you want to do with the results. For example, the survival analysis for cancer samples is one of the major analysis function people doing cancer research. So inside the Nexus Copy Number, there's another function called Nexus DB. So it's a cloud-based repository of Nexus projects. So you can get the redundant and secure backup and archive your projects and deposit into the repository inside the software. So it's a cloud-based, so it's not going to use your own local storage, and it's going to put up there in the cloud. You can also do query against your own projects or public data sets. For example, the ISCA data set, if you do constitutional samples, you can actually, based on the sample phenotype or even copy number events, you can query. I'm going to show you actually an example when I do the live presentation. Also, there are so many other public data sets we uploaded into Nexus DB to allow the user to download them for analysis. It's very easy, single mouse click. You can download a large data size, data project. For example, the TCGA, the Cancer Genome Atlas data sets. It's all up in the Nexus DB. So it's just one mouse click. You can actually download data. You can even mix your data with the TCGA or other data sets you downloaded from Nexus DB. You can also share your projects with your collaborator using Nexus DB. So that just gives you all those functions inside Nexus DB. Now I'm going to show you the software itself. It's having a live demo here. So the software, once you open, it's 3 big squares. It's a new project. You can start fresh, or you can open existing project you already processed. You want to revisit the data again. Nexus DB, as I mentioned, just previous slide, you can see here you can get into Nexus DB. Now I'm going to show you a workflow. How do you start from fresh? Once you get the data from Affymetrix Axiom CNV summary tool, how do you analyze the data in Nexus Copy Number? So you first create a new project. You give a name of the project. You can call that Axiom project. And you say, I save somewhere on the desktop, for example. You can click this, put somewhere in a folder. And then you can choose what organism you're studying. So like, as I said, we support so many different organisms from cow, dog, cat, pig, human, mouse, rat, or yeast, zebrafish. So all the things, depending on what you study. So the data I have is the human. So you pick the genome build 37, create a project. Once you have the project, the first thing you do is to load the data. So you go to the load data. You would pick the Affymetrix Axiom data. So as you can see, we support all kinds of data. So here, one of them is called Axiom CNV. So you pick Axiom CNV, then ask you the data mosaic sample, if it's cancer sample or non-mosaic sample. You can pick either one depending on your data. And the stringency is just for the software to call the copy number based on the stringency of the setting you want to do. And also you can do the correction for the waviness, as I mentioned. So if I select the data, this is the one plate of the Axiom plate, 96 files, 96 samples. To save us time, I'm going to just show you, let's say, load 5 samples. You can load all of them. You can load hundreds or thousands of samples in one single batch. So once you load in the data has been displayed here. You click done. Data will be loaded. Yeah, it's doing the analysis right now, so it's pretty quick. And here we go. So you got the results. So once the data is loaded, then it's automatic. So it's pretty simple to the users. So you load in the data, the software is going to take over and give you the results right away. So as you can see here, we have a trisomy, and it's not entire chromosome. So on the full Q, you get a gain. There's the 2Q region. You have a gain. There's some other gain, which I'm actually to open a project for the entire 96 plate. As you can see here, the same thing. I just want to save time. Earlier I did loaded 5 or 6. So now we have 96 samples. And here you can see we have all the data in here. The status says has been processed. It's the CNV, Axiom CNV data type. The quality score, like I mentioned, those are the QC metrics. So the smaller the data, the number, the smaller, the better quality of the distribution of the data. As you can see here, you can even sort by the quality. So those are the good quality data. So the very small quality score number. So as you go lower, you get only one sample is at the borderline. So we usually have a 0.2 as the soft cutoff. So anything higher than 0.2, we say the data is a little noisy. So below 0.2 is good. So you can see this one sample is on the borderline, but all the other samples are pretty good. And here you can see I have 3 other columns which I added using the so-called add factor function. You can add factor, you can load factors. The factors are actually the phenotype or the clinical factors for each sample. So you can do as many columns as you want by simply doing add factor. Sometimes people have those information already. They simply load the factor from the file. So we'll populate this table with all those columns with those values. For example, here we have gender, you have female, male samples. You can also have description of the samples. I only annotated 5 samples in this case. As you can see, the Wolf, Hirschhorn syndrome, Turner syndrome, trisomy 2 and 21, translocation, anything you can put in to describe your sample. And also I put sample conditions as abnormal or normal, so based on the copy number profile. As you can see, in these results, we have the genome view of the all the 96 samples. You just scroll down, quickly scan through what's going on in each sample. For example, you see the red color, there's the copy number loss. The blue color is the copy number gain regions. And also, since it's a SNP array, you also get error H call, like the yellow below the red. And you get a purple that's usually comes with the copy number gain. Sometimes you can even get copy neutral error H region, which you don't have any copy number changes, but you do get error H call. So you scan through the entire region. Like here, we have a error H. There's no copy number change. This is the copy neutral error H. So you can actually easily identify using the filter function, which I'm going to touch upon later. But right now, as you can see, you can quickly scan through what's going on with your plate. What kind of events, big events like this one, trisomy 8, and there's trisomy 12, then you can easily identify. So, for example, if you want to get into one single sample case, like in this case, we have a trisomy 2, you open this sample B6. This is called a single sample drill down. So you actually open this one sample. The sample info tells you how many events in this sample, how many one copy gain, how many one copy loss, what's the total copy number changes, and also plus the sample factors we put in. You go to the overview, you actually see the ideogram of the events for all the chromosomes. Like here I said it's a trisomy 2 and plus a trisomy 21. You can also go to the whole genome view to see the distribution of your log ratio probes. Like here, this plot is a log ratio plot. You see here we have a 0,9, which normally that means the copy number equals 2 regions. Then suddenly you saw this big event go up on chromosome 2, and meanwhile, you look at the lower half is the BNL frequency plot. Actually, for the normal region, copy number equals 2, not only you get a log ratio 0, you also get 3 bands in the BNL frequency plot. While you get a copy number gain, you get a 4-band pattern. Because that's a 3 copy total, you get the SNP call would be AAA, AAB, and ABB, and BBB. You get a copy number gain. Same thing for the trisomy 21 region. You can see we get a 4-band pattern very clearly. And also the other ones, you usually get 3 bands, except here you have some event. You see there's a trail going on in the BNL frequency plot. You can also go to each chromosome, look at what's going on with these changes. You can change the chromosome number right here to chromosome 2. And then we see the trisomy chromosome 2. You can actually see there's a blue color bar on the entire chromosome. That indicates a copy number gain in this region from copy number 2 to copy number 3, 1 copy gain. And actually the interesting part of this event is there are 2 copy number loss region actually inside this trisomy 2. If you're zooming into this region, you actually can see there's a copy number loss highlighting in the red color, and also there's a smaller copy number loss too. So based on the information Affymetrix provided to me, this two actually has been identified also by cytoscan to validate this copy number loss, those two. So initially didn't pay attention, and then once you look into this distribution of the log ratio, it's actually very clear. You can see this bunch of probes actually way below 0.9, and same thing here, you can see. So Affymetrix run the same sample on cytoscan, actually confirmed this copy number loss. That actually gives you the idea how good the Axiom data is for copy number analysis. Now you can go to actually the whole genome, the ideogram view. You can also click the chromosome. For example, want to go to chromosome 21, you just click it, then actually go into chromosome 21. Here, like I said, it's another trisomy situation. 3 copy total. You see the full band, clear full band information band pattern in the biennial frequency. So if you want to identify this gain, you can easily click it, and then actually this table will highlight this region. So that's chromosome 21, starting from here, there to here, and then we get a copy number event, CN gain, and give you the size, where's the cycle band, and how many probes in there, and what's the probe median value, which is a log 2 ratio. And here you can even modify this table to actually include more information to describe this event. You can have the gene symbol. You can also include decipher syndrome, for example. Then apply, you get an extra column. So right there. If you sort by that, and then click this again, you see here we have CN gain. It's the early onset Alzheimer. That's overlap with the decipher syndrome. Actually, I have another example here. This example, BOA. We have a copy number loss on chromosome 4, as you can see here, very clearly. You can easily click them to actually identify the distribution of the log ratio. This is the preview without going into the sample if you want to check what's going on with this event. Of course, you can open this sample like I did earlier for the other sample. You can see the chromosome 4 has a big loss region at the tip, and then you can actually go in there and look at this copy number loss plus the error H call, because you only have one copy of the chromosome region in chromosome 4, and here you should see beautiful 2-band pattern in the biennial frequency. That means you only have one copy because those are either A-call or B-call. You lose the middle band. Like here we have 3 bands, you only have 2. And if you zooming in, I actually include the decipher syndrome track in here. So that has this syndrome, Wolf Hirschhorn syndrome, which actually identified for this sample before even people run the sample. They already suspect this patient has this syndrome based on the phenotype. Once you run this in Axiom, you actually identified this copy number loss. So if you click the track, you actually can go to the Decipher website and give you a description what's going on with this syndrome, what kind of phenotype this syndrome usually present, and perfect match to the patient situation. As I mentioned, we have Nexus DB. Nexus DB has a lot of public cases you can query against. Let me get into Nexus DB, which requires a login, which you can get from us. So let me get into Nexus DB, logging into it. As you can see, we have visible projects. We uploaded lots of projects for people to use, either download or query, depending on what kind of study you want to do. So here you can see, if you sort by that, we have a lot of TCGA, that's for cancer research people, a lot of TCGA data. And also, you can see we have the ISCA database, that's for the constitutional people. Like here, of the ISCA almost 17,000 cases in this project, which I already checked, which means I want to query against this project based on what I have here is the copy number loss. So if you right click, you can actually query database, which will give me how many cases actually in the ISCA 17,000 cases overlap with this sample's copy number loss on chromosome 4. So we'll go through the query and give me the results. So in this case, we identified 182 samples out of these almost 17,000 cases. So if you go into all the sample call, you can actually see those are the cases. They even have notes and annotations. So if you sort by annotation, classification, you see a lot of benign, but those benign samples are basically at the very tip, has some loss. This gray area is actually what I have here, is the entire region of my case. So if you scroll down, you start to see more events, like those events are all pathogenic. So the big events overlap more with my cases. In here, a lot of them is actually related to that Wolf-Hirschhorn disease. So it's give you the description what's going on with this. So this is the easy way for people to query against public database from your case. So since our samples here, really each sample is not related to each other. So what I have here is a bunch of samples. They all have their own events. You can see here we have a gain. This one has trisomy 18. This one has some gain of 12. But you do see on the top, we have a frequency plot. So you can see 50%, 100%, and here 0, that's for the gain, that's for the loss. You can see the peaks. So those are the tiny peaks, focal gain or loss region. So if you have all the sample have, lots of sample has the same deletion or same gain, they contribute to this peak. For example, here we have a very high peak, 56% of my sample has a gain. So if you zooming into this region, actually see that gain is actually overlapped by the Toronto database DGV right here. So many samples show these gains. They all overlap right here with this peak. But you can see that's nothing unusual because it's the DGV reported region. If you click it, you actually see a lot of cases has been reported in this region, either gain or loss and gain together in that study. So you have, say, for example, 139 gain, 18 sample show loss, so that overlap with this. So this tiny gray area is actually what we have here. So inside this gain region. So this is another way for you to identify if those gains are really pathogenic or just common human CNV polymorphism. So that's the peak. As you can imagine, if you are doing some other related sample study, for example, cancer samples, you run on a plate of Axiom array plate, 96 sample, then you can see a lot of overlapping regions. For example, the breast cancer sample, you have a HER2 duplication. You can easily see that peak on chromosome 17. There is a HER2 duplication. In most of the samples, you can see that same gain. But since my sample here are not really related, so I give you the idea, so how you do this. But since I made up some factors here, like the sample condition, abnormal and normal, you can actually do this kind of study based on your sample. You can choose factors, choose the sample condition, and then you can view them based on the factor aggregates. Instead of individual sample, I can view them in groups. So in this case, as you can see, 40 normal samples, basically they have some focal gain and loss. I guarantee you all those events are the DGV overlap region. And 56 sample I call abnormal because either they have a trisomy 2 or 21 or somewhere, or I have a big gain. You can see here, you can have something 3 or 4% of the sample had that gain. So that's basically coming from probably 1 or 2 samples from there. And you also have loss. But this is not a good sample plate for research, but I just show you the idea what you can do. Based on this, you can actually do the comparison, easily compare them. So what are the regions that significant different based on the condition? For example, normal versus abnormal, you can actually generate a comparison. Means sample normal versus abnormal. So if you view them, you actually get the significant different regions based on the normal, abnormal. As you can see, as a principle, you can actually do any comparison between phenotype you have in your sample. You can do that. And let me switch back to individual samples. As you can see, and here one of the kits basically has nothing going on except those tiny DGV regions, gain or loss. But suddenly you see on the chromosome X, there's the copy number loss. And meanwhile, the Y is also a copy number loss. So actually, this sample is a Turner syndrome example. It's labeled as a female, but it only has 1 chromosome X. So it's a 45X Turner syndrome. Unlike the other ones, like the first one, BO8, we have 1 X chromosome, but the Y chromosome is normal. So this one is actually a male sample. Same thing for this sample is a male sample. And the typical female sample, you see the Y loss, but not the X. So, but this one is a Turner syndrome. You lose 1 of the X for this female. So that's another one. You can actually open it, look at the chromosome X region. Yeah, as you can see, the entire chromosome is just been lost. One of the copy has been lost. So it's here you see the log ratio is below 0, and also the biennial frequency show you typical the 2-band pattern, as I mentioned earlier. As you can see, Nexus copy number can actually handle large sample size. As you can see, for the Axiom data, we loaded 1 plate. You can load as many plates as you want. For large, like biobank or those large research centers, they handle large sample size. As you can imagine, you can actually run lots of Axiom plates, and then you can load into Nexus in even a single study. And then you can look at them together, and this become actually your local database. You can actually query even against your own data. For example, you pick this loss, you can actually show similar cases in your data, but in this case we only have 1 sample similar to that. If you do this, you see how many cases I have. Yeah, only 1 sample overlap. So I'm just going to stop right here. Just give you idea what's the strength of this Nexus copy number. What can you do downstream? A lot of functionality I haven't even touched in this presentation, because those are related to the research, and we have, like I mentioned, survival study and enrichment analysis, all those things depending on what kind of research type you have. So here we have a single plate with all kinds of clinical copy number events. You can easily identify them by going through all of them. So I'm going to stop right here, try to see if there are any questions I can address. Here is one question. For the genome that are not colored in blue, deletion, purple gain, and orange neutral, what is the copy number for them? So actually the blue is copy number gain, like I mentioned here. Purple is the 4-band pattern for the biennial frequency. So there are actually only 4 different colors. The purple, like I mentioned, blue is for the gain, red is for the loss, yellow is for the ROH. Like in this case I mentioned, there's a copy neutral ROH. Actually, I can open this one, E01. You can see we have a region on chromosome 7. There's no copy number change, labeled in yellow. You look at them, that's a 2-band pattern coming from here, biennial frequency. The copy number log ratio-wise is normal. So that's labeled as arrow H. So if you click it, then it's called arrow H and give you the size. So to answer your question, basically we have those 4 different colors. So as I said, the purple is based on the copy number gain region. Like here, we have a copy number gain on chromosome 1. You get a full band pattern. Then we get the purple color right there. So if it's yellow, that's copy number. The biennial frequency only has 2 bands, as I mentioned. You can see the whole genome view. Here we have a 2-band on chromosome 7, 4-band on chromosome 1. So that's purple, that's yellow. The gain is blue, red is loss. Hopefully that I answered your question. Here's another question. Can I mix Axiom data with my cytoscan data in the same single project? Yes, you can. As I said, in Nexus, you can load data as long as they have the same genome build. So since cytoscan and the Axiom data both using genome build 37, so as you can see here, you simply go into load data, and instead of using Axiom, you actually choose the Affymetrix cell file. You can load in the cytoscan data. Depending on if you're dealing with cancer or non-cancer sample, then you can actually do the correction for the cytoscan and for the wavelengths. Then you load your cell files into the same project of the Axiom data. If you really want to do that kind of research, you want to have all kinds of data together, and then you want to maybe the same sample you run on cytoscan and on Axiom, you want to analyze them together. That's doable too. All right. Another question, where to download the white paper or detailed description of the methods used in Nexus 7? 7.5, that's fine. So you can actually go to our website, biodiscovery.com. You actually can see a lot of white paper and even blog post, and also the even past recording of the webinar, some other videos. You can actually find a lot of information to learn about the methods in Nexus 7.5. Also, if you're using Nexus 7.5, the user manual is also a very good resource for you to learn a lot of those information. Here, another question. I'm using the Nexus Express for OncoScan. Can I log in the Nexus DB? In the Nexus DB, are there data for specific type of cancer samples? So for the OncoScan data, actually you can log into Nexus DB, but the problem is a lot of those TCGA data, for example, in Nexus DB, they are not using OncoScan. They use the old data type like SNP60. So you cannot download the data into your own project because your Nexus OncoScan software is not a full version of Nexus 7.5, does not support the Affy old array like SNP60 or Cytoscan, the new one. They only support the OncoScan. That's the answer for that question. And are there data specific type of cancer samples? Yes. As I mentioned in Nexus DB, let me show you. Inside, we actually have just curated the TCGA data. I can sort by this. You can see we have all kinds of TCGA data. We have a curated data called the TCGA Premier data, all different type of TCGA cancer samples. So like the PRADE, the GBM, the ESCA, or breast cancer. So you can actually access the TCGA Premier group. You have to join into that group before you access the data. All right. Does Nexus also support all kinds of Illumina arrays? So this is, of course, the answer is yes. So the Nexus software supports all kinds of array platform. Since today is the Affymetrix joint webinar, so I'm focusing on Affymetrix array, but it does support all other arrays, including Illumina, including the NGS data for copy number analysis. All right, so it looks like those are all the questions we have for now. So I'm going to stop right here. So if you have further questions, you can keep typing in. But before we drag this in for too long, I'm going to open the poll questions. Please provide some feedback by answering those poll questions. Poll must be closed. Okay, this is the question number 1. Okay. So the second question. Third question. Okay. Last question. All right. Thank you very much for attending today's webinar. So if you have any questions, you can still ask. So let me check. No, I don't see any questions either for me or for Mary Jo. And we have, if you have any other questions, you can still type in after the webinar is closed. So please check back. We have monthly webinars to present to you. So thank you again for attending today's webinar. Bye.